This is Chapter 8, Photosynthesis. Photosynthesis is the source of all food for all living things. It produces the glucose that all living things need to turn into energy for life. That photosynthesis, in turn, comes from the sun. So in a sense, all energy, all life comes from the sun. This is the chemical equation for photosynthesis. You don't need to know all the C6H12s. You just need to know that carbon dioxide plus water go in, glucose, which is C6H12O6, plus water plus oxygen come out. The balancing numbers aren't important. It's what goes in and what comes out. This process is called carbon fixation because we have gaseous CO2 coming in, the carbon in carbon dioxide, combining with water to become the solid sugar glucose, C6H12O6. That means that gaseous carbon has been fixed into a solid molecule. This process is carried out by certain types of bacteria and algae called the cyanobacteria and the cyanoalgae. Cyano, C-Y-A-N-O, stands for blue-green. So the blue-green bacteria and the blue-green algae can carry out photosynthesis because they are filled with chlorophyll, just like the chloroplasts of land plants. Chlorophyll is what allows photosynthesis to happen because chlorophyll can absorb the incoming sunlight. Photosynthesis occurs in three major steps. The first happens when the chloroplasts actually capture the sunlight energy. They then change that energy into energy molecules that the plant can use. Those are ATP and NADPH molecules. ATP molecules are energy for immediate use. NADPH are molecules for energy use later. What happens for the NADPH molecules to be created is the chloroplasts have to take the NADP molecule and add a hydrogen ion to them. When NADP plus hydrogen occurs, NADPH is created. This is called a reduction reaction, or you would say that NADP has been reduced. I know it seems weird to add something to something else and saying you reduced it, but this is half of what is called an oxidation reduction reaction. You'll learn more about that in chemistry. Right now, you just need to know that the addition of a hydrogen ion is called reduction. Step 1 and Step 2 must have sunlight to occur. These are light-dependent reactions. In Step 3, the energy from those energy molecules, the ATP and the NADPH, will be used to create glucose. This is the carbon fixation portion that I mentioned on the previous slide. This reaction happens in the dark, or it doesn't need light to occur, so it is called a light-independent reaction. This is a visual to show you what we'll be discussing in this chapter. On the left-hand side is the light-dependent reactions, the photosystem 2, the photosystem 1, and the electron transport chain. This is where the ATP and the NADPH molecules will be created, and you can follow them across to the right side of the screen. The blue circle, the Kelvin cycle, is step 3, where the glucose is actually made. If you follow it back across to the left side, you'll see the NADP and the ADP molecules. These are the leftovers after the energy has been used. They're being sent back to the light-dependent processes to be re-energized. This is the structure of a chloroplast, the outer membrane, which all organelles in eukaryotic cells have, the inner membrane, which is left over when the chloroplasts were aerobic bacteria millions upon millions of years ago. Remember the endosymbiotic theory. The green stacks in the middle are the thylakoid. The stack itself is called the granum. The individual green circle is the thylakoid. This is where a lot of photosynthesis is going to take place. And the stroma, which is the goo that provides shape for a chloroplast, just like cytosol does, and it will also provide a place for some additional chemical reactions to happen. As I said before, light is the most important part of photosynthesis. Light moves in both waves and as separate bundles of energy called photons. Waves are why we see light in different colors, why we see blue and green and red. And photons are individual little packets of energy that help to explain the energy transfers within photosynthesis. Molecules in the chloroplasts absorb the energy from the different wavelengths in different ways. These molecules are called pigments. Green plants use two different types of pigments, chlorophyll and carotenoids. The chlorophyll is primarily the color, and the carotenoids are where the photons are going to be. The energy of a photon can remove an electron from a molecule. This is called the photoelectric effect. The movement of these electrons through the electron transport chain is the basis of energy transfer and the formation of ATP and NADPH in photosynthesis. There are two types of chlorophyll, chlorophyll A and chlorophyll B. Chlorophyll A is the main pigment in plants and those cyanobacteria and algae that I mentioned earlier. It absorbs violet, blue, and red wavelengths. When violet, blue, and red is absorbed by our eyes, we see green. That's why the majority of our plants are green. Chlorophyll B is called an accessory or secondary pigment because it absorbs the light that chlorophyll A does not. So it's going to absorb the blue wavelengths closer to green and the reds and the oranges and the yellows. If you combine those two together, the total amount of the light that comes into a plant is increased, and so photosynthesis becomes more efficient. The carotenoids absorb the photons, so they add additional energy to the photosynthetic process, but the photons are used primarily to help move electrons down the electron transport chain. I'm going to be talking a lot about the electron transport chain. The job of the electron transport chain is to slow down the sunlight, particularly those photons and the electrons that those photons release. Because if you don't, the energy is too intense, it's too fast, the plant can't use it. The electron transport chain, through a series of small reactions, is going to slow that energy down so that the electrons and their energy can be used by the plant. In addition, during those chemical reactions, hydrogen ions will be released. Those will also be used by the plant to create energy. The combination of electrical energy from the electron and the creation of the hydrogen ions makes what is called an electrochemical gradient, and that will be vital to the creation of the ATP molecule. The light-dependent reactions of photosynthesis are going to generate energy through a process called photophosphorylation. What we're going to do is we're going to use light energy, that's the photo part, and add ADP and phosphate molecules together, that's the phospho part, to create ATP. That's the phosphorylation part. So ADP plus phosphate using light energy to create ATP is photophosphorylation. This energy creation will begin at the electron transport chain, and I'll talk about that in the next slide. Before the electron transport chain can begin its work, it has to get an electron. And that electron comes from the reaction center. The reaction center is a special chlorophyll molecule that is able to absorb enough energy from the sun to energize an electron to move it to the electron transport chain. When we're talking about photosystem 2, which is where this entire process will begin, the reaction center is a chlorophyll molecule called P680. P because it's a chlorophyll pigment, and 680 because it absorbs light at the wavelength 680. When we're talking about photosystem 1, the reaction center is called P700. All other pigments that are found in and near the reaction center are called antenna pigments. That's because they collect energy and then they bounce it to that specific P680 or P700 molecule. So they act like antennas of energy. Together, the energy from the antenna pigments and the energy that the reaction centers absorb on their own will be enough to begin the electron down the electron transport chain. There are two ways that the chloroplasts will create ATP through photophosphorylation. We're going to begin with the non-cyclic form. In this case, energy comes into the plant through sunlight, through the leaf, and into the chloroplast. The first thing that happens is a water molecule is split. The hydrogen ions will go to the interior of the thylakoid, called the lumen. The oxygen will move to the end of the electron transport chain where it will wait, and the electron will be moved to the reaction center. Because we're going to photosystem 2 first, the electron will go to P680 as the reaction center. P680 and all of the antenna pigments that surround it will give the electron enough energy to move it to the top of the electron transport chain, where a molecule called the primary electron acceptor will be waiting. That primary electron acceptor molecule will guide the electron into the electron transport chain. The electron moves down the chain in a way similar to a marble bouncing down stairs. At the bottom of each stair, a small chemical reaction will take place. In that chemical reaction, the electron will lose some of its energy. Some will go into the activation energy needed to create that chemical reaction, and some will be lost as waste heat. Remember, I told you this is really important because the electron has too much energy in its original form to be useful. Also, with each chemical reaction, a hydrogen ion is released. That hydrogen ion will move to the lumen where the hydrogens from the water are already waiting. Eventually, the electron will move its way down the electron transport chain to the oxygen that's waiting at the bottom. It will bond to the oxygen and release a new electron. The oxygen will go out of the cell as waste. The electron then will move to photosystem 1. That reaction center, photosystem 1, is called P700. We're going to leave the electron at P700 for the moment and go back to the lumen where the hydrogen are waiting. We've created a concentration gradient because there are more hydrogens in the lumen than there are outside. They're going to flow from high to low, from the lumen and through the inner membrane of the thylakoid by way of the transmembrane protein ATP synthase. ATP synthase is an enzyme that is going to create ATP by bonding ADP and phosphates together. The ADP and phosphate molecules are found in the membrane of the ATP synthase. When hydrogen flows through the synthase, it will create the hydrogen bonds and the energy needed to bond ADP and phosphate together. So hydrogen goes into the synthase and ATP comes out. Now we'll go back to that electron. Remember, it was sitting in the reaction center P700 of photosystem 1. It's going to be energized using the energy of P700 and the surrounding antenna pigments. It's going to move up into the electron transport chain and do the exact same thing that it did in photosystem 1. It's going to move down each stair step, be involved in a chemical reaction, lose some of its energy, and produce a hydrogen. The difference here is that the hydrogen don't move to the lumen. Instead, they stay near the electron transport chain. At the bottom of the chain, a molecule called NADP is waiting. The electron and the final hydrogen that was produced will be bonded with that molecule. They will produce an NADPH molecule. Remember, NADP plus hydrogen is called a reduction reaction because hydrogen has been added to the molecule, and that's called reduced. This NADPH molecule is the equivalent of 3 ATPs. It's called a final electron acceptor because this is the last stop of the electron. This is a one-way trip for this electron. Once it's attached to the NADP molecule, it's done, and it has to be replaced by incoming light. The Golgi body will come and pick up the ATP and the NADPH molecules and take it to the rest of the cell where it's going to be used. When they're broken up, the ADP and phosphate and the NADP molecules will be returned to the chloroplast and to the thylakoid so that they can be reconstructed once again through this process. The second form of photophosphorylation is the cyclic form, and this is a much simpler process. First of all, water isn't split into hydrogen and oxygen. The incoming sunlight goes directly to the P700 reaction center of photosystem 1. The electron is energized, it moves to the primary electron receptor and moved into the electron transport chain. Once it's in the electron transport chain, it acts just like it did in the non-cyclic form. Each time it moves down the step, a chemical reaction happens, the electron is slowed down, and a hydrogen is produced. These hydrogen are moved into the lumen like photosystem 2. They gather all up, they create a concentration gradient, and they move through ATP synthase just like photosystem 2. The difference here is that because we have only a single electron transport chain, when the electron gets to the bottom, it's returned to the P700 molecule to be re-energized and sent back into that transport chain again. So it goes around and around and around. If you notice, there is no NADP molecule waiting at the bottom of this chain, which means that there isn't an NADPH molecule made. The reason for this is because in this particular situation, the environmental conditions are not ideal, and there's not enough nutrients and materials to make that NADP molecule. So the plant makes the energy it can through ATP. This is not an ideal situation because not as much energy is made, and the plant only does this when it has to. It much prefers to do the non-cyclic form because more energy is created. The next two slides are a review of photophosphorylation. This is a visual showing the water splitting and the hydrogens and oxygen moving to where they belong, and the electron moving through photosystem 2's first electron transport chain. It shows the movement of the hydrogens into the lumen and the electron then moving into the second transport chain. And then the final picture shows the hydrogens moving through the synthase to create ATP. In the light-dependent cycle, water is broken down. Oxygen is created as a waste product. The hydrogen ions move through the thylakoid's membrane, creating a concentration gradient to create those ATP molecules, and the NADP molecule is reduced to create NADPH, which is also an energy molecule. Remember, NADPH is the equivalent of 3 ATPs. Now we're going to go to the light-independent portion of photosynthesis. This is the portion of photosynthesis where the glucose molecule is actually being made. That's the synthesis part. It's going to happen in the dark. It's going to take place in the stroma rather than in the thylakoid. And the process is called the Calvin cycle. The purpose of the Calvin cycle is to fix gaseous carbon dioxide into a solid sugar. It's also called C3 photosynthesis, and it's going to happen in 3 steps. The first step is the actual carbon fixation part. We're going to begin with a 5-carbon molecule called RUBP. It's going to combine with carbon dioxide to create a 6-carbon intermediate molecule, which will immediately break down into 2 3-carbon molecules called PGA. An enzyme called Rubisco helps to make this process happen faster. In step number 2, ATP and NADPH molecules that were created from the light reactions are going to be used to reduce those PGAs to G3P molecules. Remember, reduce means to add a hydrogen. In step number 3, those G3P molecules will be used in 2 different ways. Some will go to the regeneration of RUBP. Basically, we're going to break them back down into that original 5-carbon sugar to keep this process flowing. The other G3Ps will be held until the process is repeated. It takes 3 CO2 molecules to create 1 G3P molecule. So that means this process has to happen twice. 2 G3P molecules will be added together to create 1 6-carbon glucose molecule. The regeneration step also requires the use of ATP. Together, when all the steps are added up, it takes 9 ATP molecules and 6 NADPH molecules to create a glucose molecule. That Calvin cycle energy is continually being regenerated through the creation of the ATP and the NADPH molecules in the light-dependent portion of photosynthesis. This is a visual of the Calvin cycle. It shows carbon dioxide coming in at the top, being converted to PGA and G3P molecules on the right, and leaving the cycle as glucose at the bottom. It's also showing those G3P molecules being converted back to RUBP on the left, and the cycle beginning again. As we've talked about photosynthesis, we've talked about chlorophyll, we've talked about chloroplast, we've talked about thylakoids. We have not talked about the stomata. The stomata are equally important to photosynthesis because they allow carbon dioxide and water into the leaf and oxygen and water out. This exchange of gases and water is essential to maintain homeostasis. During the summer, when it gets very hot, the plant will protect that homeostasis by closing the stomata. The plan is to keep dehydration at bay. This works for a short amount of time. The problem occurs if it happens over a long period of time, because if the stomata are closed, carbon dioxide cannot get in and waste oxygen cannot leave. At night, remember, the carbon dioxide that is brought in is converted into glucose. If the stomata are closed, carbon dioxide does not come in. The Calvin cycle, however, continues with whatever carbon dioxide is still stored within the cell. Again, this works for a small period of time. If the stomata stay closed for an extended period of time, the amount of carbon dioxide in the cell gets to be too low, and the Calvin cycle can't use it. The Calvin cycle can't stop, though. It needs to be continued, so the cell has a backup plan. If you remember the first step of the Calvin cycle, RUBP combines with CO2 to create two PGA molecules. When the CO2 levels have dropped too low, the RUBP will combine with waste oxygen, creating one PGA. This process is called photorespiration. If you remember, those two PGAs will become two G3P molecules. One of those molecules will head to glucose production, and the other will be regenerated back into RUBP. When only one PGA molecule has been made, the cell will send it back to make RUBP instead of making glucose. Because remember, the Calvin cycle has to continue. So that PGA keeps going back to RUBP and around and around and around, and so no glucose molecules are made. If this continues for a long enough period of time, the plant will use up all of its stored glucose and will eventually starve and die. There are three types of photosynthesis. C3, which is typical photosynthesis, which we've talked about through this chapter because they create 3 carbon sugars at the very beginning. C4 and CAM. C4 plants convert CO2 into 4 carbon molecules when the weather is hot. This allows them to create CO2 in the chloroplast even when the stomata have been closed during photorespiration. The C4 plants can also use traditional C3 photosynthesis when the weather is nice. The CAM plants are able to do everything that the C3 and the C4 plants can do, and they also open their stomata in reverse, meaning that they open it at night when it's cooler to allow the CO2 to come in, and they are able to keep it closed and allow that oxygen to be recycled. So they have all of the benefits of C3 for cool weather, C4 when it's hot, and the ability to control their stomata when it's extremely hot, such as in the desert. And this is the end of the photosynthesis chapter.